Hello, my name is Xenophon Papadimitris. I'm a professor in radiology and biomedical imaging and biomedical engineering at Yale. This interview is part of a collection of conversations with experts in medical software from academia, industry, and regulatory agencies, and is supplementary material for a Coursera class Introduction to Medical Software that will appear in the fall of 2021. Our guest today is Dr. Nicholas Petrick. Dr. Petrick is the Deputy Director in the Division of Imaging, Diagnostics, and Software Reliability at the Office of Science and Engineering Laboratories. This is part of the Center for Devices and Radiological Health at the U.S. Food and Drug Administration. And so Dr. Petrick is talks about a variety of issues having to do with the evaluation of medical software and medical devices. And I also like to thank him. Previously, he lectured from my class. He also lectured for us at Yale. So I've gotten to know him a little bit over the years. So Nicholas, welcome. Maybe introduce yourself a little bit, tell us a little bit more about who you are, your background, and what does your division do, what you guys do at the FDA.
Sure, that'd be great. So yeah, as said, my name is Nicholas Petrick. I actually have a PhD from University of Michigan in electrical engineering systems. And so my background has been in medical imaging and started out doing signal processing for positron emission tomography types of detectors and gamma ray imaging systems. I've subsequently started working in medical imaging for FDA, as well as doing artificial intelligence and machine learning applications in medical imaging. So I work for the Center for Devices and Radiological Health. We're one of the centers within the U.S. Food and Drug Administration. Devices actually are quite a large percentage of submissions to the agency. The mission of CDRH is to protect and promote the health of the public by ensuring the safety and effectiveness of medical devices and the safety of radiation emitting products. So beyond doing medical devices, we also regulate all radiation emitting products. That's lasers and microwaves and anything else that comes along those lines. So some of it's medical, but much of it is commercial products as well. We have about 1,900 employees in CDRH. And we regulate about 18,000 medical device manufacturers and about 185,000 medical devices. So it's a pretty broad scope, and we have companies that are major international companies that are producing medical devices down to individual people or universities or garages that start with a new device concept and build it from there. So we have single or few people companies all the way up to thousands and thousands of people. As was mentioned, I work in the Division of Imaging, Diagnostics, and Software Reliability. We're part of the research arm of CDRH. And my particular group laboratory works obviously in medical imaging, and artificial intelligence, machine learning is another big area that we operate. We conduct laboratory research across a wide variety of imaging and diagnostic devices, including medical-based AI ML. So a lot of our work in my particular subgroup is in artificial intelligence and machine learning. So we have different program areas within our division. We do artificial intelligence, machine learning, that's one area. We do what's called credibility assessment. So we do a lot of work in silico modeling, where we're, instead of trying to test the imaging equipment, say what we're trying to do is build in silico models for that equipment, for the imaging and the physics that go on, so that we can understand the various concepts of those systems, as well as develop different approaches for doing assessment. So ideally, we might be able to do some of these studies in silico instead of having to do them on physical systems or on real patients. So that's one of the areas that we're working on. We also have a big effort in digital pathology. That's a relatively new area of transitioning from looking at microscopes into doing things in the digital domain. And again, has a lot of challenges because of color and other aspects. And of course, it's quite high resolution imaging as well. So we have a program there. We have another program in just medical imaging in general. We do a lot of X-ray types of imaging, ultrasound, and so forth, where we do different types of diagnosis and then deal with more sort of advanced types of imaging, spectral imaging and things along those lines. And then we have a program in mixed reality. So we're looking at how to take information, maybe those are the medical images, and then use those, say, effectively in an operating situation where you might have a mixed reality headset that's being displayed on the patient as you're doing surgery or things along those lines. So how to utilize that? How would you evaluate those types of devices? How do you determine safety and effectiveness? So that's sort of an overview of what we look at in our division and the type of work that I work on.
Thanks. So as we were discussing, we worked on the outline for this. So we'll start by looking at what you would look as from your regulator hat on as opposed to your research hat on when you get a new device in. And let's start with, maybe the simplest case potentially. So let's take a breast screening device, a piece of software that's breast cancer screening, say for mammography. And let's say this is from a company that already has a product on the market that does something similar. So this would fall under the 510K process. So how does it look like? What would it look like from your end? How do we go about it?
Sure. So let's go back a little bit and we can just talk about what we're calling software as a medical device, and this what we call SAMD. But software as a medical device is software that's intended to be used for one or more medical purposes that perform those purposes without being part of a hardware device. So something like what we call a computer-aided detection and diagnostic tool may fall under a SAMD in the sense that it's an independent piece of software or an algorithm that's applied to medical image data. And that could be applied to different vendors, mammography images and so forth. So if we fall within that range, we have products in that category already. As was mentioned, mammography, computer-aided detection diagnostic systems are already on the market. And so that can be, you would have a predicate for that device. So if we're talking about class 2 devices, then what you're going to do is compare for substantial equivalence, and we would use something called the 510K pathway. 510K is just the process of doing a comparison to determine something substantially equivalent to a currently marketed device. So in this case, if we have the company that already has an algorithm on the market, say they're coming in and they're trying to claim that they'll have better performance with that device. It'll have better sensitivity, it'll detect more cancers, and say it'll have fewer false positive marks than a predicate device. So they have a device on the market, they can do a direct comparison with their device. What we would typically do there is we would have separated the data into what we call standalone testing or an independent testing of the algorithm itself. So how well does the algorithm perform on its own? Because these are computer assist devices, the clinician is integrated with it. The algorithm doesn't work all by itself, but the clinician uses that information to make a clinical decision. So typically we'd have some sort of clinical assessment of the clinicians and determine that, when they use this algorithm, they're going to perform better than they would have either with the other device or with standard of care. In this case, because they've already done that because they have the predicate on the market, we'll tend to concentrate on that standalone testing performance. And really what that is there's going to be a data set that's acquired of images from patients that are representative of the population in the US. And again, that's somewhat of a tricky concept of what that data should look like, how big should it be, how many sites should the data come from. But ideally, it would be a relatively wide data set with a large number of cases that has a lot of different representations from subgroups and different types, both of cancers, imaging systems, as well as patients across the US. If that data set's available, and again, it would be independent of the testing part of the algorithm or the algorithm development process. And then what would happen is that would be referenced, somebody would truth that, determine where the cancers are, what type of cancers they are, what are normal patients and so forth. Once that's done, then the algorithm can be applied to that data set and determine, what's the sensitivity or false positive rate for that algorithm. Or in the case of what we typically do with algorithms, we'll look at the whole performance curve. So we'll look at what happens, not just at where it's operating, what the threshold on the output is, but across the whole range of thresholds of outputs to determine what's the curve or the ROC or the FROC curve, or receiver operating characteristics, the ROC curve, or free response receiver operating characteristic curve, FROC curve, for that evaluation. Once we do that, there'll be statistical hypotheses, obviously, of what is trying to be compared. Is it better? If you can show superiority for that data set, then we would typically sort of use that as the reference for how you would evaluate something that has a predicate, and we would use that standalone testing alone. We wouldn't necessarily need to go to the clinical step or testing case unless there were some very large differences between the types of detections that were being identified between the two systems. Again, that's a very, very simplified approach of the sort of basic data that's required. There's obviously a lot of additional data and different types of testing that would be required in a submission, from software information and documentation, the verification and validation testing for that software. There may be human factor testing that's appropriate to be done and so forth. So there's a number of different types of testing and so forth that would go on there. But the basic idea around what we look at is that standalone testing. So we'd really just be looking at the algorithm itself, being able to do a direct comparison on the same data set to the predicate algorithm and determining, is that better or not? And assuming that it comes out to be better in a statistical and clinical sense, we would probably move forward with a substantial equivalence determination.
So would you expect them to run the data set with both algorithms, like you have two arms of a study effectively in the same way that you do a drug and a placebo, and then demonstrate that one is about the same or better, I guess that's the standard here.
Right. Yeah. So typically for, again, talking about these artificial intelligence machine learning algorithms or these CAD algorithms, computer aided detection or diagnostic algorithms, we would typically want to see head-to-head running on the same data set. You know, it's possible if you had large enough data sets and robust enough data sets that you could run them on separate data. That's a little bit more of the way a drug trial is run, is you have two populations, one that gets it, one without, but they're relatively large and randomized controlled clinical trials that allow you to do those assessments. So of course you could do those for AI devices, but they're much more complicated and take more energy and time. So what we typically would do is try to do head-to-head competitions where you're doing exactly the same data set. That avoids some of the difficulties in making sure the populations match and so forth. Because a lot of these alternate effects, you know, you have shifts in population could actually produce differences in performance that would be associated with the populations and not necessarily the algorithm. So, and a lot of what we do is we try to focus specifically on, can we do head-to-head comparisons on the same data sets? And if we can do that, we can get a much better comparison with less data associated with it.
So one of the issues here, if you have a human in the loop, so this is a computer-aided type process, do you have the same human look at both data sets, or do you have different humans randomly use different algorithms as part of their aid? Because that's a big part of this evaluation.
So there's a whole structure around doing the clinical testing that we use something what we call a multi-reader, multi-case study design. So this is typically what we're using for at least medical imaging-based artificial intelligence algorithms. In this case, what we typically do is have the clinicians that are participating read in both modalities. So if it's possible or straightforward, you'd like the same clinician to read the cases in both modalities, you know, with, you know, and if you're doing comparing to two algorithms, then they would read with the new algorithm and the predicate algorithm, usually in different settings and so forth, so that there's some control over washout and so forth. Sometimes we have something like a second reader type where the algorithm is going to be applied after the standard of care is already done. In those cases, we can use what we call a sequential design where the clinician will read without the aid, make a decision, the aid will be turned on immediately, they'll make another decision or adjust their decision if they decide there needs to be a change based on the AI. And again, that has this very tight control between what was read on the unaided side to what's read with the aided side. And that helps a lot to minimize, again, the size of these studies and to improve the statistical efficiency of the trial. So with these multi-reader, multi-case trials, what we'd like to do is have a group of readers or radiologists in the case of, let's say, a mammography AI that read cases in both modalities. They're very well, it's a very controlled study in the sense that we know how many cases are being read. We try to equalize the number of cases between the readers. They're reading both images in both modalities, so that controls the correlations within those sets as well. And then we have really well-defined statistical tools for assessing. In these types of studies, there's two types of variability. There's the variability which from any study would be associated with the cases or the patients that come in. The other part, though, is in clinical reading especially, is the reader variability, is that there are differences between clinicians and how they read cases. And that variability is actually can be pretty large. So we need to account for both of those components of the variability in our statistical analysis. And so if we do these controlled multi-reader, multi-case designs, we have good statistical tools for accounting for both types of variability and then how they interact with each other. They're not just independent sources of variability, but there are interactions between them. And we can use those tools to do analysis.
Okay, so let's move now to a different device. So in the previous case, we had a predicate. There was an existing device on the market that had already gone through the regulatory process. Now, this is a new application. We can pick one. You had mentioned some optical applications. We'll get there. So now this one does not have a predicate, so it's evaluated on its own. And I assume we're still in a class 2, so we're probably using something like the DeNovo process. So how does this differ from the previous process? What are we looking at here?
Yeah, so if we had a new device, again, we could take an example of sort of an optical device that may be something like a digital pathology system or something like that. Nothing on the market, there's no predicate for it. The agency may decide that, you know, we can figure out how to do controls for that device that we think it could be in class 2. So we think the risk isn't that high, and we think we have ability to set up a structure around what we call special controls that allow us to have a structure around assessment for that device that will make us confident that we can put it in a class 2 and we can do comparisons to that device like we would. So it would become a predicate for any future device in that class 2, and we can move forward. So we've done that a lot with a lot of artificial intelligence and machine learning. Not necessarily every AI ML will fall into that category, but in things like radiology and so forth, we've gone through that pathway. Likewise, we just had a DeNovo we did for a colonoscopy or gastroenterology type of AI, which is a computer-aided detection system. And that fell, again, was decided that we can write those special controls we could put in class 2. So in this case, there is no predicate. So we don't have the clinical sort of study. So if we're again talking about a computer-aided, say, detection, so assist device, so the clinician's still involved in the diagnostics, we would follow a similar pathway. We would do that standalone testing because what we want to do with that testing is to benchmark the performance and make sure it meets its performance criteria. So there's some criteria that set out, we think it can produce this amount of sensitivity or this number of false positives and meet those criteria. So we do the standalone testing to benchmark performance. The other reason we do standalone testing is because when you put it out in the clinic, what the clinicians want to know is how well should I expect this to perform? How many marks should I expect to see on a case? Or what's the sensitivity specificity of this device? So that they can sort of gauge how to use it when they're using it in practice. So that would be the first part that would fall pretty similarly to what we did in the predicate device, although there wouldn't necessarily be a comparison to there's no predicate to compare to. The second phase is then that clinical assessment. And what we want to do now is assess not the device itself, but the clinician. How well does the clinician perform when they use that device? And so again, we would use, as we talked about before, usually a type of multi-reader, multi-case study design where we would acquire data, the clinicians would read it without the aid and then with the aid. And that could be in different sessions. It could be within the same session, depending on the type of AI that it's being implemented and actually how it's supposed to be applied. Is it a second reader or is it a concurrent read? Is it a first read type of device and so forth? And then once we do that clinical study, the goal would be to show typically that it's better than standard of care. So there's no predicate here, so we're not going to directly compare to the predicate, but what we can compare it to is what's the standard of care that's happening now. So how well do those clinicians perform in standard of care? And when you add this aid, do they do better than that standard of care in a statistical and clinically significant way? And if you can show that, then, again, those are the two main pieces of the types of studies we would do. There's lots of details and nuances of how you would set those up and what's appropriate, the references and how you truth things and things along those lines that are really important. But the basic concepts are those two basic studies are used as the main data sources for determining whether it's a safe and effective product. Again, there'd be lots of other potential types of testing on the software, human factors, other types of areas that might be appropriate, electrical safety if there's some sort of computer or something that's important. So those are pieces that would come along with it as well. And assuming all those are worked out, then you would determine that this device is safe and effective, and then we would grant a DeNovo is what we call it, where now it becomes the first product in that class 2, and other devices can use it as a predicate. And now we just follow the standard pathway we talked about the first time around.
So you used thinking about my students here, used the expression special controls as part of that discussion. So maybe you could talk a little bit about general controls and special controls and what are these things for class 2 devices?
So general controls are the basic authority we have in the medical device laws. So things about adulteration, misbranding, device registrations, those rules that apply to those that you need to do, pre-market notification, records and reports that you need to keep, the good manufacturing practices that are available. So those are sort of the general ideas of in the law, these are our regulations, you need to follow these regulations around these various aspects. And if you do that, then if that was all that was needed, then we would say that this device could be marketed. So something like a class 1 device, which are these lower risk devices, those would fall under just general controls. If you follow the general controls, most of them are exempt from pre-market review. So the device, if it's in class 1, follow the general controls. You don't need to submit something, you can market that device. Special controls are used when general controls aren't good enough, that we can't be satisfied safety and effectiveness with just general controls. So they're usually device specific and includes things like pre-market standards, post-market surveillance requirements, patient registries, special labeling, and then potentially pre-market data requirements. So that would mean there's some type of testing or data that's required pre-market in order to evaluate that device. So those, we call them special controls because they're related typically to a device, a type of device, which is in class 2. So in class 2, you typically have general and special controls, which are these specific data and requirements associated with the device class.
So let's just touch briefly on class 3 here. So all of the software we've talked so far, our screening algorithms, our CAD algorithms, were class 2. What would make a piece of software move into that higher category, which is under stricter scrutiny, I guess?
Yeah, so there are two sort of basic areas which would move that software up. One is the risk associated with how it's being implemented. So what is the disease or condition that it's applied to? So is that a life-threatening disease and condition? And then what's the type of information that software is providing? Is it, diagnostic? Is it therapeutic? Is it making determinations about how to treat or how to control the patient? And so the more that you're into these areas where it's making therapeutic decisions and it's associated with, say, a life-threatening disease, the more likely that software would move, from class 2 to class 3 types of applications. So typically, class 3 devices are life-sustaining devices, implantable devices. So if for whatever reason your software was part of an implantable device, that would likely move it into class 3. And then new devices or novel products that come onto the market where we don't know good ways of developing sort of specific special controls for those. They may be too new or have too different of approach where we're just not sure how to deal with them, and we would put those into class 3. That way, when each system comes in, we're going to evaluate each system on its own. It's not compared to a predicate or a different product on the market. It's reviewed on its own in its entirety, determined that itself is safe and effective. Sometimes we start with a device in class 3, and then after a while we get confidence, we understand how to evaluate, we get some additional testing, and then we can down classify that to a class 2 type of device. We do that a number of times with imaging devices. One example was full field digital mammography. It originally was a class 3 device when it first came in. Eventually it got down classified to a class 2 device once we had a little bit more experience about what type of testing was necessary for that.
So we'll transition now to more technical part of the discussion, having sort of gone through more of the regulatory, and we had 2 or 3 top different things we want to talk about. You had mentioned false positives and sensitivity, and you typically use, receiver operator characteristic curves or ROCs for evaluation. Let's talk a little bit about that and how you use those to define endpoints for studies. Thanks.
Okay, so we were talking about how we do assessments, and so when we're talking about computer-aided diagnostic systems, usually what we're talking about is the sensitivity and specificity of those systems. So if you have a patient that comes in and you're trying to decide whether that patient has cancer or not, you would say on this patient, the AI said it was cancer. Patient does have cancer. That's part of the sensitivity assessment. There's another group of patients that don't have cancer. You're trying to measure the specificity, or how often does the AI say the patient doesn't have cancer when they don't have cancer, in that population. So that's typically the operating points we think about, or that people think about a lot. What ROC as a receiver operating characteristic curves are this whole trade-off between the true positive fraction or sensitivity versus the false positive rate, or the 1 minus specificity. So this is 1 minus specificity. And that's for all the various thresholds that the AI algorithm may use. So typically in an artificial intelligence, you have an output score that comes out of the algorithm. So for a case, you say it gives a score on a 0 to 1 scale, with say 1 being higher, meaning it's more likely to be cancer. At some point, the company or the developer decides, you know what, I'm going to make a threshold at 0.75. So everything about 0.75, I'm going to call cancer. Everything below 0.75, I'm going to call not cancer. So that would give us this sensitivity, specificity, operating point of the device. But what we're interested in is that overall performance of the algorithm. And so what we can do is plot the sensitivity and false positive rate for all the different thresholds of that algorithm. And we can track that across the whole spectrum. So we can go from 0 to 1 in false positive rate, what subsequently goes from 0 to 1. So here's an example of a classifier. Typically, when we have a distribution, we have, again, this non-disease distribution and the disease distribution, and they overlap in some extent. So we're not going to get perfect separation between these two for most classifiers. So we have some overlap. If we separate those two out, again, put this threshold on there, we can get a single operating point. If we start and just sweep across the whole range, we can get the full ROC curve. And so this ROC curve tells us more about the fundamental properties of the algorithm and not for just one individual operating point. So this is important because sometimes we have trade-offs in device performance where we have, say, a device A, which has a sensitivity specificity operating point of here, and a device B with a sensitivity specificity operating point here. And in this case, you can see that this device has higher sensitivity, but it also has lower specificity. So the question is that a good trade-off of performance? Is this operating point better than that operating point? Now, we could do something like utility analysis and try to determine what the value of a true positive, false positive, and so on. But another way of doing this is to actually look at the ROC curves. So are these two ROC curves the same or different? So in this case, the device B actually falls on a lower ROC curve, which gives us an indication that overall, this technology probably isn't as good as the technology for device A. Now, it's possible it could have fallen on a higher ROC curve, and it would have shown that that device was better. So again, this is a way to look at the technologies more of in a fundamental properties and try to determine which of these technologies are providing better information. Because in general, you can control the operating point. We can adjust these thresholds and operate anywhere along this curve that makes clinical sense. And just as a follow-up, one of the metrics we use for doing the assessment here is not just sort of looking at the ROC curve, but looking at the, say, the area under the ROC curve. So now the performance metric, which is a combination of sensitivity and specificity, is this area under the ROC curve. And so the higher the ROC curve, the more of this space it takes up, the better the performance of that algorithm is. And then I'll just finish up by saying one of the nice advantages of this is we have a lot of statistical tools for assessing this type of ROC curve and doing statistical evaluations between two systems. And so those are helpful when we're evaluating performance.
We had talked about before is that you had mentioned you're doing some interesting research on sort of phantom and physics-based testing for images. I mean, in some ways, when we have physical systems, we don't always have to do the statistical approach. We can actually look at the properties. So let's talk a little bit more about that as well.
Yeah, so one of the other areas that we work on is really thinking about imaging systems and what type of testing is necessary for those systems. So, one of the areas we can always do is we have a new system that comes on the market. We could do some sort of clinical assessment of that system and determine whether it is, better than a predicate device on the market. That's difficult for a lot of imaging. So think about a CT system. What types of applications do you do CT for? Well, you do it for the thorax. You look at the lungs. You could look at the belly. You could look at the colon. You could be, looking at the head. You could be looking at bones in the legs and other areas as well. So there's lots of reasons you do a CT. And so trying to determine that in a clinical context, this CT is better than another CT that's on the market for every application is a very complicated, difficult task. So what we try to do is again, move things back away from that clinical data and say, what types of information would tell us about the performance of the system? So can we do, say, phantom experiments? Can we do physical measurements? So what are the physics properties like modulation, transfer function, and noise properties of the system that we can measure? And then we can then evaluate those systems. And if we look at the physics and say the phantom properties of the system, we may be able to determine, is this sort of in a physics sense equivalent or better in some way than the predicate devices on the market? And that gets us away from trying to sort of evaluate every possible clinical task that might be appropriate for an imaging device and talk more about the fundamentals of what that system is, how does it operate, and is this adequate enough to be on the market and be comparable to others. The one thing I just want to keep in mind too is substantial equivalence doesn't necessarily mean that it's superior in every way to a predicate on the market. There may be some trade-offs. Some parts of it may be a little bit better, some parts may be a little bit worse, but overall, is the overall substantially equivalent to what's on the market? And so again, there's lots of these other properties. Sometimes we get higher resolution imaging systems, but they have some more noise properties. Is that a good trade-off? There may be situations where it is. There's also a lot of complications in imaging systems, especially three-dimensional, around the reconstructions and the algorithms that are used as well. And again, those impact the various properties of the system.
I mean, when you have a lot of systems, you mentioned reconstruction here, just my personal curiosity here. There's also a fair amount of parameters that you can use to tweak those algorithms and those reconstructions. So does the validation happen over a sweep of those parameters, or is it like, this is the recommended setting, this is what we're going to use? Because
yeah, so for some types of fundamental physics properties, we would look at the physics of the system and not necessarily try to engage into all the various ways you could do the reconstruction. So what we'd be interested in is more the fundamental. What is the ability to acquire the data with the system, knowing that how you do the reconstruction has an impact. In other scenarios, we would be looking at the reconstructions directly. You know, if we're looking at some types of enhancement or so forth on lesions or things along those lines, and the reconstruction is an important parameter for that. Often we try to do sort of what we call task-based assessment. So we may just take typical tasks like a detection of a lesion, so a sort of a spheroid object. And again, when we do something like that, we may do phantom studies of that and determine how well the machine can detect those various objects. Mammography is a good example of something where you have a phantom that has both sort of blob-like masses that, you know, are sort of associated with lesions, as well as very sort of high intensity or calcification-like objects that are very small but higher in contrast. And we're trying to do both of those tasks. So how well does the system detect both of those different types of objects, which are, you know, in some sense, a task related to detecting cancer in mammograms.
Okay, so transition from an area where we understand the physics and we have a hope of understanding how it works, to an area we are all learning about, and this is sort of this whole explosion of deep learning and machine learning applications where we are all struggling to understand what these algorithms do and how to evaluate them. So I'd like to hear a little bit of what the thinking is around this. I'm sure you get lots of submissions. Everybody is trying to use AI and ML, and this is the buzzwords here. What are the fundamental issues here? What are the challenges that you're seeing as trying to evaluate these algorithms we don't understand, as opposed to the physical devices where we can get a handle on the actual physics and design phantoms to go after them?
Sure. So one of the areas one of the things I would just say is it's important that we try to figure out how to understand them. And, you know, I completely understand that that's not the case right now with a lot of deep learning, where there's a lot of art to putting these algorithms together. They're very complicated as far as number of layers and structures and feed forward and feedback loops and so forth and integrated within them. But the more we can understand why these systems might be operating the way they are, the easier it's going to be to try to evaluate them and understand, and then come up with various testing that may not be so much clinical testing on large populations, but more specialized testing to determine that they're consistent or they'll work appropriately. So that being said, we still have to live with these algorithms that are complicated and we don't quite understand. What we've done is, I think, and we've been working on these for more than 20 years, is to have a structure in place for all of these statistical learning or machine learning types of algorithms. So regardless of whether it's a deep learning algorithm or it's, you know, a linear classifier with features, they can be evaluated in the same way in assessing the standalone data as well as the clinical populations in the data. Now, the deep learning algorithm may be more complicated. It may have more variability associated with it. There are differences in properties, but the assessment methodology that we've laid out for AI in general is still applicable for how we approach it. And the biggest question is, what types of data do you need? So now the question becomes less about the structure of the testing, but then what is the data requirements? What type of data? How large does it need to be? What distributions, what subgroups need to be within that data to give confidence that it'll work appropriately in the clinical population? And that's a challenging question. We always are trying to balance the, you know, complexity and difficulty and cost associated. Well, we don't deal with cost, but the complexity and the difficulty associated with acquiring data and getting quality data with how much do we really need to show that this device is, you know, substantially equivalent if there's a predicate, or is safe and effective if it's operating on its own. And that's a big challenge. And we're evolving in our thinking of how to do that. What types of subpopulations should be reported on? What's the data we need within those subpopulations? And then this is also tied into the claims of the device. What is the device really trying to do? And that's another factor that always has to be thought through here is you could have exactly the same algorithm, but make two different claims on what it's doing, and you would have different data and different information that we'd want to see as far as how we go about assessing that algorithm. So starting out with, what do I want this to do? How do I want it to operate? Should it work autonomously? and be a diagnostic on its own, or should it be an aid to the clinician? Is it giving a piece of information, or is it the fundamental information for the decision? Those all matter in how you set up and how you use that device, and then how you might go about testing it.
And in general, I mean, this is talking to people on the other side of the regulatory question, device makers. People are able to communicate with the FDA and ask questions and in some ways get information as to what the testing procedure should be, right? There's pre-submission queries and things like that help.
Yeah, sure. So we have a lot of mechanisms. One is what we call the Q sub process, where a company can come in before they've done a formal submission and ask us questions. So they come in, they give us enough information that we can understand what questions they're asking and try to address those questions. And those could be very simple. I want to think about this is my device. Is this appropriate predicate? And we can give them feedback on whether that's the correct predicate, or whether we have other predicates that might be better, or we don't think there's any predicates at all for that device. Or they could be questions on the study design or the testing. We're expecting to do this type of testing with this study. Would that be appropriate for evaluating the device? So that's sort of a formal, what we call the Q sub process. We also have things like guidance to documents online. So for certain types of devices that say, if you're this type of device, this is what our current thinking is around how assessment would operate. It's not the only way potentially to do it, so there are alternatives to it, but basically says if you follow this type of approach and provide this type of data, this would be appropriate for a submission of that device. So those documents are online. And occasionally there's more informal interactions. We go to meetings, we present ideas and concepts on assessment methodologies and physics testing and other things along those lines that are other opportunities for people to learn about what we're thinking and ask questions about some of the methodologies that we're using, both in our research, but also that's propagating into regulatory science and what happens in submissions.
Is there anything else you wanted to bring up that I failed to ask, or is there something you wanted to say?
No, I just would say that, one of the main parts of any device development is thinking through, first of all, what you want that device to do in the clinical context. So always sort of keeping in mind, not just what the technology you're trying to develop it is, but how is it going to be utilized in the clinic. And that really is one of the most important aspects to always think about in device development. Because you could develop sort of a perfect AI algorithm, but if it doesn't have a good context in the clinical use, it may never be used. And vice versa, you may have a modestly performing algorithm or a system or device, but it's put into a context where there isn't very much there. And so that added value is really important in clinical context. So really thinking through that aspect is one of the key parts of doing a good device development for something that will end up being utilized when it's effective. And then the other part is as you have questions to talk to the agency, I think we try to be as open and as approachable as possible so that we can give you good feedback and avoid having lots of rounds or miscommunications on what's submitted. It's difficult sometimes when you get a submission that provides really the wrong information because that will either end up with an NSE or a non-approval for that device, and the company will have to start over again. So trying to do the right study up front should streamline the review process and make it a little bit more efficient, and hopefully your device will move forward into use on the market.
Nick, thanks for doing this. Thanks for taking the time.
No problem. I appreciate it.